Hi everyone, thanks to the organizers and thanks for being here today. I'm going to be talking today about hybrid allelic imbalance and gene expression evolution in the Anopheles-Gambi complex. So the Anopheles-Gambi complex is comprised of 8 recently diverged species, and as species diverge, they accumulate genetic incompatibilities that can result in postzygotic reproductive isolation, meaning that these incompatibilities inhibit intergression between species. This manifests itself in F1 male sterility in hybrids between these two species. And that's because males of Anopheles are heterogametic. They have XY sex determination, and that's different from Aedes mosquitoes. So according to Haldane's rule, hybrid sterility will first evolve in the heterogametic sex because there's only one copy of the X, and any deleterious mutations on the X chromosome have to be expressed in the males. So genetic divergence can have a large impact on the hybrid phenotype, and in particular postzygotic reproductive isolation when regulatory regions are involved, or regions that are upstream of a gene that impact its regulation. And divergence in these regions can result in gene misexpression in the hybrids when they're not interacting effectively in these F1s. So as I said, Anopheles-Gambi mosquitoes have heterogametic or heteromorphic sex determination, which means that dosage compensation operates between males and females. And so dosage compensation means that you either have double the expression of the X chromosome in males or halving of X chromosome expression in the females to balance those genes that need to be expressed in both sexes that are not sex biased or sex specific. So in Drosophila, dosage compensation takes place through hyperexpression of the X in males. In C. elegans, there's a halving of X chromosome expression, and in some mammals, there's in mammals there's X chromosome inactivation. We know that dosage compensation operates in Anopheles-Gambi. This is a paper from Rose et al. What they did is they measured X versus autosome expression in males and females, and they compared that to expression ratios between the second chromosome and the third chromosome. What they were looking for was to see if those were both 1-to-1 ratios. And they found that with increased RPKM thresholds, so this is a measure of normalized gene expression, those two ratios converge, showing that dosage compensation does operate. And they also compared the male to female expression ratio of autosomal genes and X chromosome genes, and again found a 1-to-1 ratio. So we know that dosage compensation exists in Anopheles-Gambi, but we still don't know whether it's a hyperexpression of the X in males or a halving of the expression in females. So this led us to a few questions. Does hybridization impact dosage compensation? Is a disruption of dosage compensation the reason that we're seeing F1 male sterility? Does hybridization result in the misexpression of genes? Do these misexpressed genes exhibit allele-specific expression? And that means the preferential expression of either the maternal or the paternal allele of that gene. And does hybrid misexpression result from divergence in cis or trans acting elements? A cis acting element would be one that is upstream of the gene, so something like an enhancer region or a transcription binding site. Transcription factor binding site. Whereas a trans-acting element would be one that's further away on the same chromosome or on a different chromosome. So we can measure whether or not divergence in gene expression in hybrids is cis or trans-acting by comparing the expression of the parental strains to the expression of the hybrids. If the expression in hybrids is out of bounds, essentially, of what we find between the two parental species, we would attribute that to trans-regulatory divergence, whereas if it's misexpressed but still within the range of the two parental strains, we would attribute that to cis-regulatory divergence. So in order to address these questions, we created F1 hybrids between Anopheles glutzi and Anopheles arabiensis in both directions of the cross. This allows us to observe the effect of the X chromosome in males, because depending on the direction of the cross, they're going to inherit only one X chromosome from the maternal strain. For all of my diagrams and everything in here, the maternal strain is first and the paternal strain is second. So what we're doing here is we're analyzing gene expression in Arabiensis and Kalutzii males and females, pure strain, and then both directions of the Kalutzii Arabiensis or Arabiensis Kalutzii cross. So for example, males of the Kalutzii Arabiensis cross have the Kalutzii X chromosome and lack the Arabiensis X chromosome. So in order to map this data, we created strain-specific pseudogenomes by mapping the RNA-seq data from the parents back to their species-specific genomes and incorporating variants of the parental strains into the genome. And we went through a few iterations of that and eventually came up with pseudogenomes based upon the pest coordinates and created cDNA pseudogenomes based upon that. What that allows us to do is map transcripts back specifically to the genes and then assign them as to whether they came from the maternal or the paternal strain. We used Bowtie to map the hybrid data to biparental pseudogenomes by giving each read the chance to map to either pseudogenome. And then we used a program that implements a Bayesian method called ASTMH to assign each transcript to either the maternal or the paternal genome. The data that I'm going to show you to begin with is based upon total gene expression. It's not parsed into maternal versus paternal reads. What I've done here is similar to the Rose et al. Paper. We're analyzing male versus female expression ratios at the top, Anopheles arabiensis and Kalutzii, and then males and females of both directions of the cross. Our expectation here is that on the left, X-linked genes will have a 1-to-1 ratio in the males and the females if dosage compensation is acting properly. And these are the autosomal genes for reference. What we find is that in both parental strains, this ratio centers around a 1-to-1 ratio, showing that, yes, in both Kalutzii and Arabiensis, dosage compensation is taking place. In these bottom graphs, the ratio is from the male to female expression ratio of the male to its paternal strain, and in the females, it's the male to the maternal strain, and then the paternal strain to the female. What we find is the ratio of gene expression from male to female expression for both X-linked and autosomal linked genes, the distribution is the same, although it differs from a 1-to-1 ratio. And so this shows us that in both the male and the female hybrids, we're getting kind of hyper expression of the genes, but it's happening on both the autosomes and the X chromosomes. Next, we looked at the ratio of X to autosome expression across all of the males and females of the parental strains and the both directions of the hybrid. And we didn't find any significant deviation between any of these crosses or the parental strains. This is a log 10 scale. And so we would expect all of these means to center around 0. Of course, we're going to have genes that are sex biased or will fall outside on the margins of that distribution. But on average, we're seeing that dosage compensation is taking place. And this is just a test to show that there's no significant difference between those distributions. Next, we used the program DSeq to look at log fold change and differential expression between all the treatments. And we pulled out the 100 genes from females and males with the highest expression ratio variation between the libraries. This allows us to pull out some that we can see are misregulated in the hybrids. So for example, we have some sets here that we can see are species specific, and we have some that are in red upregulated in the parental strains in comparison to downregulated in the hybrids. These blocks are similar across both directions of the female cross because they've inherited two X chromosomes. All right, so their genomes, irrespective of the direction of the cross, are the same. Whereas males, we can see a lot more variation, and that's due to the effect of the X chromosome. If we do a gene ontology analysis of this gene set, we find overrepresentation of genes involved in DNA replication, DNA binding, chromatin organization and organelle organization, and for protein classes, histones. So of course these are involved in DNA packaging and also transcription. So next we're going to look at the allele-specific divergence, and the goal of this is twofold. We want to identify genes that are misexpressed in hybrids that show cis or trans effects. And also, this allows us to identify genes that have diverged between the parental strains, but then have conserved their gene expression level. And so we do that by plotting on the x-axis the log2 maternal versus paternal expression for the parental strains. So for example, dividing male Arabian cis expression by male Kalutii expression, and then looking at the log2 maternal versus paternal F1 hybrid expression. So we're assigning the reads to maternal or paternally derived, and then comparing their expression to their parental expression. What we'd expect is to find everything to pile up right here in the center, which shows no cis or trans divergence or any interactions between cis and trans acting regulatory elements. So anything that's on the horizontal blue line indicates that it's trans divergence, so operating from a different chromosome. Anything that is on the diagonal line would be cis, and anything that's on the center vertical line shows evidence for compensatory mutations, and that means that in one parental strain or another, you've had a cis-acting variant that has disrupted the gene expression of a gene, and then you have a trans-acting variant that modulates or suppresses that. So this is showing essentially evolution for gene expression conservation. So this is the plot as I've described for the F1 females. As I said, we would expect everything to line up on this intersection here, and any deviation from that shows cis or trans, or cis plus trans, or cis bi trans interactions. What I just want to highlight here is that there's a disproportionate number of loci that are lining up on the vertical axis, which indicates compensatory regulation of gene expression. So this is the case where in one parental strain or another, you've had a cis-acting variant that has disrupted gene expression and a trans-acting variant that is acting to compensate that. We find a similar pattern in males, but with more evidence along this x-axis for trans-acting variants, and that's due to the direction of the cross and the action of the X chromosome, or the influence of the X chromosome on autosomal expression. So these are trans-acting variants that occur on the X chromosome that are influencing expression of autosomal loci. So in conclusion, does hybridization impact dosage compensation? We find overexpression of genes in male and female hybrids in comparison to parental strains. However, dosage compensation has not been disrupted. Does hybridization result in misexpression of genes? We do find misexpression of some genes involved in DNA replication, binding, and organelle organization, and also overrepresentation of the protein class of histones in those genes with the most variance between the libraries. So these warrant some further exploration. Does hybrid gene misexpression result from divergence in cis or trans acting elements between parental species? We have evidence for allelic imbalance in F1 hybrids, as shown by cis and trans effects. A higher proportion of genes exhibiting trans regulatory divergence in males due to the effect of the X chromosome, and a large proportion of genes showing evidence for compensatory gene evolution in Arabiensis and Kalutii. So with that, I'd like to thank the members of the Anopheles Genomics Consortium for help with my many questions, and members of the Slotman Lab at Texas A&M University. And the study was funded from a seed grant from our Genomics and Bioinformatics Core and a dissertation improvement grant from NSF. Thank you.
All All right, we have time. We have time for one question. Okay, then we'll move on. Thank you. Oh, sorry.
Yep.
Have you done this kind of modeling in the debate between Anopheles cluzii and Anopheles gambia?
We have not done this in Anopheles cluzii and Anopheles gambia. That would be a great comparison to look at because of the incipient speciation question, and to see if particularly one of these regulatory variants is evolving more quickly. We have extended this comparison to Anopheles gambia and Anopheles cortlandia.